Your brain contains about 86 billion neurons, and each of them can be connected to thousands of other neurons. So that means that your brain is, well, extremely complicated. But if you think about it, in previous videos, we've talked about the fact that neurons can cause other neurons to fire. So one neuron fires, sends neurotransmitters, and that can cause another neuron to fire, or at least influence it so that it's more likely to fire in the future. But if you think about that, that kind of poses a problem. Because if neurons are just telling each other to fire, then wouldn't your entire brain, all 86 billion neurons, be active all the time? Wouldn't they just be simultaneously firing? I mean, even if they're just indirectly connected in chains and webs of neurons, wouldn't at some point that excitation cause all the neurons to be simultaneously active? In fact, what I just described is not so different from seizures. So a seizure is basically a wave of action potentials, of excitation basically coursing across the brain. Can be in a certain region, or it can be the whole brain. It can be just in one hemisphere or both. But regardless, obviously we're not all having seizures all the time. And so there must be some sort of inhibitory mechanism that keeps neurons from just constantly telling each other to fire. And in this video, we're going to be talking about exactly what that mechanism is. Specifically, we're going to be talking about the neurotransmitter GABA and how it basically tamps down the activity of neurons and tells them not to fire. And near the end of this video, we'll be talking about drugs like alcohol, Xanax, and Valium, and how they affect this system, and how that explains a little bit of how alcohol affects the brain. So without further ado, let's get into it. Hey, if we haven't met, I'm Andrew and this is Sense of Mind. This channel is all about giving you the tools to understand your brain so that you can upgrade your mind and improve your life. We do videos like this one, which are a part of the kind of Introduction to Neuroscience series, where we're going from the very small, starting with neurons and neurotransmitters, the mechanisms of how all that works, and building out to brain regions, systems, even looking at kind of cognitive neuroscience and psychology. It's going to be a long journey, but I hope you'll stick with me along the way. So in that series, we will have basically 3 levels of difficulty for each topic. So we'll have kind of a 30-second, 30,000-foot overview that just gives you really the basic ideas, the main ideas of what we're talking about. And then we'll have a longer kind of 5 to 15-minute video that explains the basics, gets more into the technicals of how whatever it is we're talking about works, in this case GABA and inhibition. And then we'll have a longer video that is dedicated to giving you a much more in-depth treatment of the topic. So whatever level you're interested in, whatever background knowledge you have about neuroscience, we have a video for you. So definitely check those out. Now, in addition to those Introduction to Neuroscience videos, we will have a kind of less technical collection of videos that are more about just giving you tools to upgrade your mind and improve your life. So we've had a video about different tools for avoiding depression and for just increasing your mental health. So check those out if you're interested. And hit that subscribe button or click follow if you're on Instagram. And make sure to like this video and comment any thoughts you have. All right, so let's talk about GABA inhibition and eventually alcohol and drugs, like I mentioned. All right, so GABA is known as the brain's main inhibitory neurotransmitter, as I kind of mentioned earlier. That means that its role in the brain is basically to tell other neurons to slow down. So we're going to get into the molecular biology, some of the mechanism of how that actually works, but just a little bit more information on GABA. So about 20% of the neurons in the brain have an inhibitory function, And I did double check that number. I had in a previous video was talking about glutamate neurons, a different neurotransmitter system, and I had misquoted that number, but this one I believe is correct. Around 20% of neurons in the brain have an inhibitory function, and this makes GABA the second most commonly used neurotransmitter in the brain, aside from glutamate. And if you need a primer on glutamate and excitation in the brain, check out our previous videos on those topics. And so you might imagine that if GABA has an inhibitory function where it's kind of telling neurons to slow down, to not fire so much, it makes sense that it would have a kind of calming effect. And actually, a lot of people do take GABA as a supplement to help them sleep or to calm them down. And as we'll see near the end, a lot of drugs that are considered sedatives or just depressants tend to work on this system. So anyway, let's get into how GABA actually functions and how it tells neurons to not fire or influences them to stay quiet. All right, so what we've got here are our sending neuron and our receiving neuron, our pre and postsynaptic neurons respectively. And let's look at these a little bit closer. Okay, so as I mentioned at the beginning of the video, we talked about the fact that if all neurons were excitatory, what would stop the brain from having seizures all the time, right? Why would that ever stop? Why wouldn't neurons just have this continuous chain, this loop of excitation going through the brain? Another problem is that in the last video we talked about glutamate, which is the brain's main excitatory neurotransmitter. And the thing about glutamate is that if it is released onto a neuron too often, if a neuron binds too much glutamate in too short of a time, it can cause excitotoxicity, which is basically death by excitation. It excites neurons to death. So what do we do about this? How can we prevent these things from happening? Well, the answer is inhibition. So first of all, I just want to note that inhibition does a lot more than just stopping the brain from killing neurons and seizing all the time. It is also just like a generally useful mechanism in the brain. If you think about a car, right? An analogy to a car, you don't just have a gas pedal. I mean, theoretically, if you just had a gas pedal, you could just take your foot off the gas to slow down. But that wouldn't give you very precise control over the acceleration of the car. So you would want to be able to both decelerate and accelerate fairly quickly. So that's why, you have a brake in your car or on your bicycle. So it's kind of the same idea with neurons, with the brain, with neural networks. So neurons are arranged into functional networks, basically networks of neurons that do things, that compute the answer to something, how to move your hand in a certain way, what you're seeing in front of you. This all has to be kind of decoded or coded by neurons. They interact in these networks, and these networks include a lot of these inhibitory neurons, often called inhibitory interneurons, that function to basically coordinate the pattern of firing within these networks. So that is kind of one of the main roles of inhibition in the brain. All right, so then what we're looking at is the synapse between these two neurons. So let's zoom into that so-called GABAergic synapse. All right, so what do we see here? Well, this is our classic image of a presynaptic neuron on the left and a postsynaptic neuron on the right. The presynaptic neuron is releasing neurotransmitters, in this case GABA, onto the postsynaptic neuron, and those little circles are the synaptic vesicles holding the neurotransmitters, waiting to release them like the one at the top just did. So just as a quick reminder, let's go through how glutamate changes the activity of the postsynaptic neuron. So Glutamate is the excitatory neurotransmitter in the brain. It is like the direct opposite of GABA. Basically, what it does is it binds, its neurotransmitter receptor binds glutamate and allows positively charged sodium ions into the cell. And that positive charge coming into the cell raises the voltage of the neuron. Right? And so that makes sense. That's what we've been talking about for the last few videos. But we want to know, how does GABA do its inhibition? And I just want to mention a couple of terms really quick because it's something I probably should have mentioned earlier. But when the presynaptic neuron sends a signal to another neuron, it can either cause that neuron to become more likely to fire, right? In the case of glutamate, it's an excitatory neurotransmitter, or less likely to fire, like GABA, like we're talking about with this role of inhibition. When the effect is to cause the postsynaptic neuron to be more likely to fire, we call that an excitatory postsynaptic potential. So it's excitatory because it's raising the likelihood that that neuron will fire. It's postsynaptic because it's on the postsynaptic neuron. And potential, that final P, is just kind of another word for voltage. So it's an excitatory postsynaptic potential. And then in the case of GABA, when it inhibits the postsynaptic neuron, that's called an inhibitory postsynaptic potential. And then these are abbreviated as EPSP and IPSP. So anyway, you'll probably come across that at some point, and I thought that would be important to mention. Okay, so we just saw how the postsynaptic neuron can become excited by glutamate. Now let's go back to GABA. Let's talk about what GABA does. So GABA has basically two different mechanisms by which it can cause the postsynaptic neuron to be less likely to fire. So the first one we'll talk about is called the GABAa receptor, GABAa, and it is ionotropic. And if you remember from the previous couple videos, ionotropic means that it has an ion channel nearby or attached to it that allows ions to flow in one direction or other when the neurotransmitter binds. So the receptor is attached to a channel, an ion channel, and when the neurotransmitter binds, it allows ions to flow. So let's see how that works in the case of GABA. So GABA floats across that synapse, binds to the neurotransmitter receptor, and that causes the channel, the attached chlorine channel, to open up, and it allows, there's a chemical reaction inside the channel that allows it to open up, and then chlorine flows into the cell. And now you'll notice chlorine ions actually have a negative charge. So you're bringing negative charge into the cell and thereby turning the voltage, the potential, the membrane potential of that postsynaptic neuron down. So you're just making it less likely to fire by introducing negative charge into the postsynaptic neuron. Okay, so that is the first mechanism. And I'll say with that, that is an example of an inhibitory postsynaptic potential, specifically in the case of the GABAa receptor, the ionotropic one that we just talked about. It is fast, both with regard to how quickly it takes action on the postsynaptic cell and how quickly it closes. So it's really like a fast break. It's just really quick and it has a fast action, but it doesn't persist for very long. And so now we'll talk about the second type of GABA receptor, which is slower in its action. Okay, so this is the GABAb receptor. This one is metabotropic. And as we talked about in the case of the glutamate metabotropic receptor, what happens is when the neurotransmitter binds, when GABA binds to it, the receptor causes a number of chemical reactions, a kind of molecular cascade of events inside the cell. That's what all those blue circles and that blue arrow are. And those affect a nearby potassium channel. And the little blue Pac-Man eating the square on the potassium channel represents a chemical reaction happening on that ion channel, which allows it to open up, right? Undergoes some kind of reaction that allows it to open up. And then potassium actually flows out of the cell. So it's kind of the opposite mechanism of the ionotropic receptor, because the metabotropic GABAb receptor doesn't let negative charge into the cell, but rather releases positive charge outside of the cell, but it's having the same net effect of turning down the voltage. And again, as I mentioned, these metabotropic GABAb receptors are slower to activate and also slower to close. So they're kind of a slower grinding type of break. I think of maybe the difference between the ionotropic receptor and the metabotropic receptor as something like the difference between the brakes on your car, like really sensitive brakes on a car, versus maybe the brakes on an old train when it's coming in and they just gotta really crank that brake to get it to stop, and it's just screeching and taking forever, and then it's harder to remove the brakes in that situation. Okay, so that is really the basics of the mechanisms of how GABA works, how it inhibits neurons, causes those inhibitory postsynaptic potentials. And now let's zoom out a little bit. Let's look at a neuron as a whole and see how this kind of plays out. Okay, so here we have our kind of graphical representation of an action potential. So on the right, we have this neurotransmitter receptor binding an excitatory neurotransmitter, and that allowing positive charge to come into the cell, and that causes an action potential at the axon hillock, which is really the base of the axon of the neuron, and that causes that positive charge to kind of course down the axon, and then eventually on the left cause the release of neurotransmitters from the tips of the axon. So that is the basic picture. But as you can tell, as we talked about earlier, neurons are connected to thousands of other neurons. And so the reality is that it's really never, or it's very rarely, that one neuron causes another neuron to fire in a kind of one-to-one fashion. The more likely scenario is that there are thousands of neurons talking to a single neuron, and they're all telling it, either fire or don't fire. They're giving it inhibitory or excitatory postsynaptic potentials, and then that neuron kind of sums all of those inputs up at the axon hillock, and then it will either fire or it won't. So it all comes together in that single event. So there's this chorus of voices talking to the neuron, and it has to weigh all those up and see, does it tip it towards firing or not firing? Now, the reason that's relevant is that as far as inhibition goes, if we look at this neuron again and we say, okay, so it's receiving inputs all over the place from both excitatory and inhibitory neurons. Is there a way that you could, if you were trying to inhibit this neuron, keep it from firing, and you could only choose one spot on this neuron, on either its dendrites or its cell body, to give it an inhibitory input to release some GABA onto it, what would be the best place? And so to kind of illustrate that, I've got these three different places you could choose. So spot A down there at the bottom right is the tip of a dendrite, like a dendritic tree at the very tip of one of its kind of far-flung dendrites. Spot B is an area of the soma, that is the cell body, but it's pretty far away from the axon hillock. And then we've got spot C, which is also on the soma, on the cell body, but right next to that axon initial segment, the axon hillock. So if you had to guess, would you say that A, B, or C would have the strongest inhibitory effect on this neuron? So if you guessed C, you'd be correct, because it is the closest to the axon initial segment, and so it's going to have the strongest influence. Because if you think about what's happening is when a neuron receives an inhibitory postsynaptic potential, especially when we're talking about that GABAa receptor, when it allows chlorine into the cell, that chlorine can dissipate, right? And if it's very far from the axon initial segment, that influence of the negative charge isn't going to be as strong as if it's very close to the axon initial segment, because that's where the action potential will be triggered if there is indeed going to be one. So that's just something to keep in mind. And there are different types of inhibitory neurons that synapse onto different parts of another neuron. So now let's talk about those drugs that I mentioned at the beginning, specifically alcohol, barbiturates, and benzodiazepines. So if you know anything about barbiturates or benzos, you'll know that they have, well, a kind of calming, relaxing, sedative, depressant type of effect. They slow you down, so it's not that weird to think that they work on this GABA system. But so does alcohol. And kind of a central question I want you to keep in mind is, if all these molecules act on the same system, how is it that they have different effects? So first, let's just see what they do in a general sense. So we've got our dendrite of our postsynaptic neuron, and we've got our GABAa ionotropic receptor, which is the specifically the type of receptor that these molecules interact with, rather than the metabotropic GABAb receptor. So what happens is these different molecules bind at different locations on that GABAa receptor. So the alcohol is going to bind in a different place than benzodiazepines, and that is going to be different from where barbiturates bind. But their basic effect is that when GABA binds to the receptor, they just enhance the action of GABA. So they just allow more chlorine in effectively, or really inhibit that cell more than it would have been with just GABA. Now, here's the question that I just posed. If all these compounds have the same general effect of kind of tamping down the activity of these neurons, then why do they have different psychological effects? Why is drinking alcohol not very much like taking Xanax or barbiturates? The reason comes down to how these receptors, how the GABAa receptor is constructed. Because the reality is that there are different types of this receptor, and because it's built from different subunits. So there's these different kinds of subunits, and the different molecules, the drugs that we're talking about, alcohol, barbiturates, benzodiazepines, are actually binding to those individual subunits or to spaces between the subunits. So if you have a GABAa receptor that has one type of subunit but not another, well, alcohol might bind to that one, but benzodiazepines might not. So it's this issue of how the receptor is actually constructed. And then what's interesting about that is that these different versions, these different flavors of the GABAa receptor are present in different brain regions. And different brain regions do different things. So it makes sense that these different drugs should have a different psychological effect. Because, if you drink alcohol, it's going to affect different brain regions than if you take benzodiazepines. Because even though they're both working on GABAa ionotropic receptors, it's these different types of that receptor that these different drugs are actually binding to. It's the subunit structure, the composition of the receptor. So let's talk a little bit about how that works with alcohol. So you put some alcohol into your brain, right? It goes through your bloodstream into your brain, and it is going to affect various regions by affecting that GABAa receptor. One other thing to mention with alcohol is that it's not only working on the GABA system. There are some other general effects. I mean, if you think about it, ethanol, if you've ever used it to clean a cut or a surface of any kind, you'll know that it's a pretty effective solvent. It's a cleaning cleaning solution, and so you can imagine that this solvent, this organic solvent that you're releasing in your body, is going to have a lot of general effects on cells. It's kind of like a bomb going off in your body to some extent, but not that extreme. And one of the things it does is it actually affects NMDA glutamate receptors. And we talked about that in a previous video, and that'll become relevant in just a minute. But just know that this is not the only thing that alcohol is doing. But let's look generally what is known about which brain regions are affected during alcohol intoxication. So you drink some alcohol, it is going to get into the brain, and these are some of the regions that it affects most strongly. So we've got at the front here is the prefrontal cortex, and this region is really responsible for what we often call executive functions, so kind of behavioral, emotional regulation, understanding and acting according to social rules, the ability to self-regulate. So there's kind of a lot of regulatory functions there, and that makes sense. People become pretty disinhibited on alcohol. They dance on the tables. They go crazy. They drive cars when they shouldn't. So that can kind of explain some of that. Also, I'd like to draw your attention to the cerebellum. So this is that little structure at the bottom of the brain, and it's actually sometimes referred to as the little brain. That's what cerebellum means. But it is really highly involved in smooth movement and other functions, but helping your body to execute smooth movements. So anything like dancing or anything where you're really moving your body in kind of a smooth pattern. That is going to go through the cerebellum. So that also makes sense. So one of the things that police check for during a DUI stop is they look at if someone's eyes can smoothly track a light, or if they can do this thing where they're touching their nose and counting or whatever it is, and they're looking for somebody not being able to execute those motions smoothly. So de facto, the police are looking for cerebellar inhibition. Basically, the cerebellum's not working as it should, and that also can affect, of course, is going to affect motor movements and including driving. And so there's some other effects going on, like in the brainstem and thalamus, which are, the brainstem controls a lot of autonomic functions, so functions of the body, and the thalamus is responsible for relaying sensory information to the cortex. And then, so we also see the sensory cortices. These are areas responsible for processing the information from the five senses. And so that also makes sense. People tend to, have distortions in their sensory world. They enjoy music more, perhaps, or they might not see as well, might not be seeing as clearly, and so maybe that has to do with this alteration in activity in the sensory cortex and or in the thalamus. Lastly, you're probably familiar with the fact that alcohol can inhibit memory formation, or at least storage. People black out on when they drink too much alcohol, meaning they don't remember a portion of the night. And that is probably has to do with the fact that alcohol affects not just the GABA system, but also the glutamatergic neurons. Basically, it blocks the process of memory formation by inhibiting NMDA glutamate receptors, which are really important for long-term potentiation, which is essential for memory formation. It's basically the strengthening of synapses between neurons. So you can see that that alcohol has a lot of effects in the brain. So yeah, that is kind of how all that works. Okay, well, that just about does it for GABA, inhibition, alcohol, Xanax, benzodiazepines. If you enjoyed this video at all, please give it a like and subscribe to this channel, or follow if you're on Instagram. And let me know your thoughts in the comments. I really want to keep this conversation going, hear what maybe wasn't clear, or what you'd like more information on, or just if there's any topics that you'd like me to cover in the future. Thanks so much for watching. This channel is brought to you by the Diamond Mind Foundation, and this video was written and produced by Andrew Cooper Sansone. Thanks so much for watching. I'll catch you next time.